I'm here today to tell you a little bit about a problem with mitosis, a very common problem that we have in our society, and that is cancer. So cancer is really mitosis gone off the rails or off the tracks. And so today I want to share with you kind of what loosely happens in cancer and what kinds and parts of cell division go off in order to encourage that particular process to happen. So up here, we have mentioned these cell checkpoints, right, are really important. There are a lot of enzymes that run these checkpoints, and the enzymes actually surge inside of the cell. So to lower these gates, remember these gates, these cell cycle checkpoints are almost like stop and go ahead flags, right, that the cell is receiving. And to lower a flag, one of the things that will surge are these enzymes like these CDKs, those are cyclin-dependent kinases or mitosis promoting factors. They'll surge in the cell and they actually lower those gates so that the cell can move forward. The cell, because of these checkpoints, actually pauses, and it has a really rigid way that it times out mitosis so that a cell isn't dividing out of control. By definition, a cell that is dividing out of control of these cell cycle regulators is a cancer cell. So that's something that we don't want to see happen inside of your cells. Now, it's interesting, these enzymes, we've mentioned this before, that proteins come from your DNA code, right? So you're coding for these, and you've received information from your mom and dad on how to code for these cell cycle regulators. And it just so happens that you're gonna inherit one copy of a particular enzyme from your mom and one from your dad. So this sets up almost like a two-strike in you get cancer kind of system inside of your cells. Cancer is any sort of cell that is dividing out of the control of these cell cycle regulators, and most of us in our lives will start our life with cell cycle regulators with two good copies of an enzyme that's regulating these particular checkpoints. Now, some of us will then acquire hits across our lifetime. You've probably heard of carcinogens, different chemicals in our environment that can cause hits or mutations in these cell cycle regulators. When you get a mutation, if you start with two good copies and you get one mutation, then you're still good because you still can make that protein product. You still have a gene inside of your cells that is functional. So as long as you don't get two hits, then you can still produce that product. However, we're accumulating hits across our whole entire lifespan. And so if you get two hits against you, right, you start with two good chromosomes, but by the time you're 65, you've received two hits in a particular cell, then that cell will basically knock out that cell cycle regulator. Depending on the regulator, that can lead to out of control cell growth. And that is really what cancer is. Now, when we look at normal cells versus cancer cells, usually I kind of do this in a Venn diagram, but normal cells, they're going to divide under the controls of these enzymes and checkpoints. They will obey density-dependent inhibition. That means they don't divide when they're crowded, right? They sort of slow themselves down. And they'll send signals to each other because your cells are always talking to each other. And they anchor into your tissues. So cells that are communicating with one another are actually anchored in, and they stay anchored. We call this anchorage dependence. They stay within their tissue. They don't float free or start to sort of go off on their own. Cancer cells, on the other hand, cancer cells ignore those checkpoints, right? You get hits in them, so they will not be pausing and timing and regulating mitosis correctly. They'll be dividing often out of control or very quickly and not pausing. They often don't pay attention to crowding situations. So as your cells are crowding, they're like, woo, let's just make some more. And third and worst is metastasis, where cancer cells start to float free. They lose anchorage. They lose that sort of dependence on those other cells and that communication, and they start to float free. And at that point, they can move into other locations of your body, set up tumors in other places. So I kind of just wanted to bring out a problem with mitosis, and I wanted to just remind you that cancer really starts with one cell that gets a hit in these cell cycle regulators. It starts dividing out of control. That's when we get a tumor. If it kind of loses this anchorage dependence, it can then start to float free into other tissues. If it's something like breast cancer, you have all this lymphatic and blood tissue that is running through your underarm. It's very easy for those cells to then migrate to other places inside of your body. When we look at preventions and treatments of cancer, preventions are things like exercise and healthy diets and seeing your doctor and avoiding these carcinogens, right, that sort of cause these hits and mutations to occur in cell cycle regulators. But when we look at treatments, it's interesting that they're going to focus on stopping mitosis often. Either you're going to surgically remove cells that are misbehaving and dividing out of control, right? So those are things like surgeries to remove cell groups or tumors, or chemotherapy is going to focus on stopping mitosis. So there are different chemotherapy drugs that stop DNA replication so you can't copy your chromosomes correctly. There are other kinds of chemo drugs that stop the mitotic spindle from growing out and attaching to the chromosomes and thus stopping mitosis. That particular drug that freezes microtubules is known as Taxol, and it actually was ripped out of a Pacific yew bush that grows in front of our music building. So if you're ever on campus, you can look for that particular building. But these kinds of treatments really target mitosis and target stopping it. So thanks again for watching my video clip, and I'll see you next time.